Okay, so today I wanted to talk a little bit about some of the statistical challenges that arise in modern neural electrophysiological experiments, and some new approaches that we've been developing to try to deal with those challenges. And in particular, we've been developing methods related to the theory of marked point processes to understand, instead of how individual neurons code for information about the outside world, how populations can simultaneously code. And I want to frame this in terms of a case study trying to understand hippocampal replay and see how the types of statistical challenges that arise in the analysis of hippocampal replay and some of the methods we've used to try to overcome those challenges. And so thanks to Ingrid's very nice talk earlier, I don't need to explain, action potentials and how they relate to local field potentials, but maybe I can just mention that modern neuroscience experiments often require a lot more integrating of information across multiple sources. Instead of a very simple stimulus response paradigm where we look at the activity of just a single neuron in response to a stimulus or to behavior. Oftentimes now we're recording from hundreds to thousands of neurons simultaneously and trying to understand relationships between the activity of all those neurons, potentially in multiple brain areas, to rhythms that are recorded in the local field potentials that integrate synaptic potentials across thousands to hundreds of thousands of neurons and relate those all to features of behavior. And this reflects I think a larger trend in neuroelectrophysiological experiments of increasing complexity, both in terms of the stimuli, the response, and the associations that we're looking to understand between them. In particular, in terms of the stimulus, we've moved largely from simple stimuli that were entirely experimental controlled, typically one-dimensional, to ones that are very high-dimensional, that are quite naturalistic, that an animal might experience in its actual experiences, that are largely uncontrolled within the experiment. And we've moved from responses that typically look at the activity of an individual neuron to responses that look at the coordinated activity from large populations of neurons and how those relate to various rhythms in the brain. And so we've also gone from looking at a single type of modality, maybe just action potentials, to responses in terms of multimodal signals. And more than any of that, we've moved to trying to understand more complex associations. For example, from understanding stimulus-response relationships that we typically assume to be stationary to ones that explicitly try to capture dynamics in the representations. And in particular, we've also moved from trying to understand directly observable relationships where what's being encoded is entirely controlled and observed, to ones that reflect maybe neural representations of cognitive features which can't be directly observed and must be modeled using some type of latent processes. So I wanted to discuss the ways we've dealt with some of these challenges in the context of a particular case study, trying to understand hippocampal replay in awake rats. In particular, if you have a rat exploring an environment and you record from the medial temporal lobe, in particular the hippocampus, people are largely familiar with the notion of place cells, where if, say, this rat here moves from the blue area through this green area to this red area, and you record from multiple neurons simultaneously, you may record neurons with place fields that are more in the blue area that will respond when the rat is moving through that blue area. Other cells with place fields in the green area, which won't respond when the animal's here, but will respond when the animal starts to move through the green area. And then maybe other cells with place fields in the red area over here, which will respond only when the animal moves through there. And so as the animal moves through its environment, what you can see is a very active rhythm in the local field potential, in this case, a theta rhythm, that corresponds with this place-specific coding. And if you look at the activity over this population, you see a pattern of spiking moving from these blue neurons through the green towards the yellow and red. And all of that is related to the behavior in terms of moving through this environment. But it's been long seen that when the animal isn't actively exploring, when it's sleeping, or even when it's awake and at rest, either before moving through these environments or after moving through these environments, what you can see, as shown in this kind of red box right here and expanded right here, is that occasionally there'll be a change in the local field potentials reflecting a sharp wave and a fast high frequency ripple event. And when that occurs, you'll see patterns that have features that are statistically similar to when the animal is moving through the environment. In this case, the animal is stationary at this point, but you see a pattern of the neuron that fires with the blue receptive field firing first, then the green, then the orange and red. And so you see similar patterns, which are temporally compressed, but don't just represent temporal compressions of the data itself. They match in some way statistical features as the animal moves through this environment. And similarly, we can see another example here in blue, where you have, again, this high frequency ripple activity. And now you see activity which doesn't actually reflect anything that you would see during actual experiments. In this case, the red neuron fires first, then the green, then the blue. But again, you have a pattern of activity that's in some way related to the population pattern that you see during active exploration. And so our goal is to try to understand these various types of replay events. In particular, our experimental collaborators, in particular, Lauren Frank's lab at UCSF, has already shown that if you interrupt all of these replay events with stimulation, then the animal has trouble learning new navigational tasks. In this case, the animal is moving on an M-shaped maze. It has to alternate from the center left and then right, as we saw in the previous talk. And if you interrupt all these replay events, the actual behavior itself never reaches the same behavior as an unstimulated animal that's allowed to have these replay events. But our experimental collaborators were interested in these various different types of replay events and whether they could determine which types of replay events were going and stimulate them based on their actual content. And so this presents a number of different statistical challenges that I want to take some time to go through. Maybe the first and foremost is they want to be able to detect the content of these replay events in real time. And so that means that they don't have time for the spike sorting step that was explained during the last talk. In particular, if we have maybe multiple neurons that are recorded on the same tetrode, those neurons all have separate waveforms, and various spike sorting techniques are available to maybe find various templates, in this case, maybe red, blue, and green, representing 3 different neurons here. To sort each of the spikes based on those templates, and then to build separate coding models for each of those neurons. So instead of spike sorting and building a separate neural model for each of those neurons, our goal is to try to directly relate the coding properties of each spike based on its waveform without making an explicit spike sorting step. And so the approach that we used here is that instead of using a simple univariate point process model for each neuron, we built marked point process models for entire tetrodes that may include multiple neurons, where we defined a joint mark intensity function that we call lambda here. It's changing as a function of time, but also has this multivariate mark that in this case might reflect things like the principal components of these waveforms, or maybe just the peaks of these waveforms on multiple electrodes. And we characterize the probability of seeing a spike in the neighborhood M of a particular mark, of a particular waveform at each moment in time. That's what we build the model to describe. And then we can use those models to try to decode the animal's either position in this environment or these replay events by looking at sequences of spikes with particular waveforms. And so here's just an example of the difference between these two approaches. So on the top is just the classic approach, looking at an individual neuron and trying to build a receptive field model. In this case, it's a spline-based receptive field model, a spline-based pace field model, where we've linearized this M-shaped track where this origin, this O point here, is the center, and trajectories that move from the center to the right are located to the right of that origin. Ones that move from the center to the left are to the left of that origin. And if you spike sort one of these neurons, you find a place field that tends to fire just before this A location as the animal moves up the center stem. And it looks like it fires more when the animal moves toward the right over here than when the animal moves towards the left. And you can conduct a test. And in this case, for that neuron, you'll find statistically significant differences between right turns and left turns. However, if we build a joint mark intensity model, in this case with the mark just being the peak height of each spike, what we see is that when the peak height is very large, we get the neuron firing with spikes that look like that about equally before turns to the left and turns to the right. But we also have some spikes with slightly lower magnitudes, slightly lower peaks, which can't be distinguished, can't be separated by spike sorting from the ones with larger ones. But the ones with the smaller peaks tend to only fire when the animal is moving to the right. And so in this case, if you actually saw the waveform itself and you had this joint mark intensity model, then you'd be able to tell if you saw maybe a spike with a lower amplitude, you'd be able to predict with higher confidence that the animal was about to take a right turn. Now, maybe going back to looking at higher dimensional marks. So in this case, looking at the peak heights from each of 4 electrodes in this tetrode, what we see is that if we build one of these joint mark intensity models, we capture simultaneously both features of the spike sorting. So in this example, I'm showing, for example, the peak height in channel 1 versus the peak height in channel 3, and the intensity, just a slice of the joint mark intensity as a function of channel maxima. And you can see that the same information that goes into spike sorting is present in this joint mark intensity model. While simultaneously, if we look at, for example, this linearized position as a function of channel maxima, then we see individual blobs that represent the place fields of each of these kind of clusters. But now one of these models is capturing these multiple place fields simultaneously. And we have multiple mathematical approaches for capturing these types of joint mark intensity models. For example, we can build non-parametric models that use kind of kernel-based estimators that just place a maybe Gaussian kernel at each observed spike location, both in terms of position, but also in terms of the marks. Or we can build a parametric or semi-parametric model, say as a mixture of Gaussians, that tries to capture each of these individual blobs with its own Gaussian structure. Okay, so that addresses potentially the first challenge of not having time to spike sort. A second challenge is that in the experiments, these representations tend to be dynamic, and they tend to be dynamic in multiple ways. The receptive fields of each neuron tend to shift. So for example, the shape of a place field as a function of position is known to change as a function of experience. These place fields often tend to become more narrow, so become more place specific. They tend to shift in their centers, and they tend to increase in their firing intensity with experience. But in addition, you may have changes as a function of marks. For example, you may have electrode drift, which leads to the spike waveforms changing through time, or you may have new marks, new clusters of marks appearing, or ones disappearing as neurons die or are electrically revealed to the electrodes. And so one of the statistical challenges is how to, instead of treating these models as stationary models, track their evolution through time. And we've come up with a number of approaches, including some simple ones, such as just including the spike times themselves as an additional mark in the model, to an approach that I'll discuss a little bit in a slightly different form later, using state space models to actually explicitly model temporal changes in these clusters. But another maybe simple way that I want to briefly discuss is a hierarchical modeling procedure where we use a Gibbs sampling method to actually track the evolution of each of these clusters through time. So in particular, this Gibbs tracker approach consists of 2 steps. So we're using our mixture of Gaussians model that I described previously. The first step is a stochastic cluster assignment where we take each spike that we observe and we, in multiple iterations, assign that spike to one of the clusters in our joint mark intensity model. And then in each of those iterations, we take all of the spikes that are assigned to that cluster and use a standard Gibbs procedure to update both the center of the model in the mark and position space, the span of the model in mark and position space, and the intensity of that component of the model. And so that can allow us to track all of those changes through time. And so I can just show you an example. So this is an animal moving through a circular maze. I'm just showing a linearized position here on the top plot, and then the peak height in terms of the first and third tetrodes, and then just snapshots of those. And then in different epochs, so the epochs are marked by these red bars here, we've tried to build these joint mark intensity models using a mixture of Gaussians approach. And what you can see if you go from this green epoch to the cyan epoch to the red epoch, if you just look at the activity and the spike marks and locations in time, it's very difficult to just see by eye changes in the population activity. But if we look at our model fits, both as a function of the joint mark intensity across these tetrodes and as a function of position, what we can see is the expansion or maybe growth of a new component that represents activity at position, maybe at about position 1 here, where we have a very small component with quite large amplitude spikes in terms of at least tetrode 3, which then expands, maybe shifts its center and expands to a broader range of spike waveforms. You know, whether that's the appearance of a new neuron or whether that's one neuron's receptive field and space of waveforms expanding, we can't say, but we're able to capture that transition using this approach. A third statistical challenge when we build all these models is how we actually determine whether or not the models we've built are accurately capturing the statistical properties of spiking, the coding properties of the neuron. And in particular, with these kind of high dimensional models, it's very easy to get models that seem like they're capturing a lot of the structure and the activity, and yet may have a lack of fit in various components. And so we've expanded upon traditionally known point process goodness of fit methods, which use time rescaling to assess the quality of fit, for our joint mark intensity models, which use a combination of both time and mark rescaling. And we've come up with a number of different transformations. In this case, I'm showing one of those, which is a joint mark and time scaling. So in this case, the SIs are the actual spike times. The MIs are the spike waveforms for each of those spikes. And we're transforming those into a set of UIs and VIs, where the UIs integrate the joint mark intensity for each observed spike waveform up to that observed spike and then normalize it. And then we also rescale all the marks and get a distribution of all the waveforms that we have, and then use a Rosenblatt transformation for each dimension of the mark in order to get a transformation where, if our joint mark intensity is correct, our spike times and spike waveforms should transform into these V's and U's, which should be samples from a uniform distribution in a D+1 dimensional hypercube, where D is the dimension of the mark and the extra 1 is the dimension of position in this case here. And so this is just an example for some of these models that I'm showing, where we have a joint mark intensity as a function of a linearized position here. But you actually have a drift in the marks through time. This is, if you sort these, you actually get activity from 2 neurons here, where both of their spike waveforms in one dimension, in this one-dimensional mark space, are drifting through time. And if you don't have a model that captures that drift through time, you can see through this transformation, that misspecification, in the way that these spikes are transformed and are not and you can use simple known statistical tests for uniformity in the cube to reject the hypothesis that these are uniform samples and therefore reject this joint mark intensity. Whereas if we build the type of adaptive model that I just described and then perform this transformation, then at least using this transformation, you end up with samples that are not distinguishable from a uniform. Okay, so that's the third challenge. The final challenge I want to discuss is how you actually invert this process of building the model in order to decode either the animal's position through time or in terms of these replay events to decode the animal's cognitive state as it maybe perhaps mentally explores positions in this compressed time frame. And so the way we do that is we use these joint mark intensity models to describe the relationship between a cognitive state process that I'll describe as X here, which describes either where the animal is in its environment, or for these replay events, is a state that represents maybe continuous cognitive paths through the environment. And so the models that I just described define a relationship between these states and a set of point process observations. Our Ys here are our spikes and the waveforms. And we add to that another state process, in this case, a discrete state process, which I'm defining I1, I2, I3, which actually not only controls the observations and how they change in time, but also controls the dynamical model of this continuous state. So for example, if we want to detect whether or not our neuron is decoding actual position versus replay, or whether that replay is a forward replay or a reverse replay, or a replay of an outgoing trajectory versus an ingoing trajectory, we can code all of that with our discrete state process. And then once we've defined that, then the mathematics for actually decoding ends up being quite simple. If we want to compute the joint distribution of our discrete state and our continuous state as a function of the increments of our point process and their spike waveforms through time, we can express that posterior distribution at each moment in terms of that same posterior distribution at the previous time, multiplied by a state model that describes how our state processes evolve through time, how the animal either moves or how it cognitively explores, with our joint mark intensity model that describes the probability of observing a spike with a certain waveform as a function of our discrete state and our continuous state variables. And so we can compute that moment by moment. And this is one example of just a decoding of that discrete state. So in this case, the black is the animal's actual locations. This is all actually very quick. It's about a quarter of a second, this whole movie. And what I'm decoding here, in red, I'm showing just the maximum a posterior estimate. And the color code around that is showing the posterior distribution of this cognitive state through time. And so what you can see is the animal's not moving at all, but in this quarter second, we have evidence in the spiking of a trajectory, a replay trajectory that moves away from the animal's location, takes a left turn, and moves up the track as well. In addition to estimating this kind of mental cognitive state, so I'm showing here just a snapshot of that. So this is one replay event. Again, this linearized position from O, the center, either left or right. And now I'm showing that posterior distribution linearized through time. But in addition to estimating that continuous state, we can also estimate a discrete state. In this case, the discrete state takes on one of four possible discrete states. It's either an outbound or an inbound replay event, and it's either a forward replay event or a reverse replay event. So it's either looking at a movement or a mental exploration of the movement in fast forward, going forward, or rewinded. And what we can see is that if we're estimating this X variable, the position, simultaneously with the state variable, after just a few milliseconds, in this case, within 100 milliseconds, we're able to determine with a high degree of confidence the type of replay event this is, as well as the trajectory of the replay event. In this case, I'm showing two replay events, both of which start at the center and move outward, either to the right or to the left. But in fact, these are quite different. This first replay event represents a outbound forward replay event. So the animal perhaps thinking about moving away from where it is in fast forward, versus this replay event, which also moves away, but is actually an inbound reverse. So the animal replaying its movement from the outside, from the left back into the center, but then that movie played in rewind. And so this gives us our first step towards potential closed loop interruption. Our experimental colleagues at UCSF have given us the challenge of trying to, within 50 to 100 milliseconds, understand the content of each replay event so that they can potentially stimulate and interfere with these replays within that threshold, that time period. And so if I require a 80% confidence in my discrete state, then in fact, for nearly all of the dozens of replay events in a particular experiment, I'm able to determine which of the 4 discrete states it has within 100 milliseconds. And the more confident I need to become, the longer it takes me to become that confident. And in black here is the fraction of replay events which I'm not able to detect with confidence. But this shows us that we're able to move using all of these approaches towards potential closed loop interruption. Okay, so I've tried to highlight a number of different statistical challenges here and give a taste of the way in which these marked point process modeling approaches can help us address each of those challenges. And in particular, these marked point processes allow us to understand coding properties of large-scale populations, assess the qualities of those models, and infer features or decode features about the signals driving the neurons. And this state space approach that I described in this fourth challenge provides the kind of link between either stimuli or behavior and the neural activity, or in a case like this, between complex cognitive processes and neural activity, which gives us the potential for these types of closed loop experiments where we're using information inferred moment by moment from the brain in order to drive our experiments. So I'll end there. I just want to acknowledge the many people who helped devise these algorithms, who actually implemented them, as well as my funding sources. Thank you very much.
Thank you a lot. Thank you, Yuri. Uri, sorry. Yeah, Yuri is in the room. I'm sorry. Who has a question or a comment? Yeah, Ingrid, go ahead.
Sorry. Thank you very much, Yuri. I'm sorry because I didn't really hear properly. So I would like to be sure to understand from the biologist's point. Tell me, with the tools, you label Gibbs, it was something GIBB.
Gibbs sampling.
Yeah. Does it mean that we can affirm that we are recording along the session the same neuron?
Yeah, so that's a good point. If over a long period of time, something, you're looking at your neural activity and it looks like it's changed through time, either maybe through different epochs of the same experiment or, over multiple days, there's the question of how confident can you be that you're recording from the same population? And, if, for example, I look at the data set from here and I look at the end, the beginning of this experiment and the end of this experiment, it seems like I have some place fields here that look about the same. So maybe this neuron here, maybe this cluster represents a single neuron here, which looks like it's the same at the beginning and the end. What this Gibbs sampling procedure allows us to do, as well as some of the other approaches that we've taken to address this problem, is it allows us to track moment by moment how these are changing. And so one consequence of that is you might be able to see maybe a change from the beginning to the end or a change from one day to the next, and you might be able to track that change through time. And so there is the potential here to be able to say that, this cluster that's changed a little bit in its coding properties and maybe even changed in terms of the shape of its spike waveforms, because of the kind of continuity of that change through the experiment, I can say with high confidence that it's still the same neuron and it's just become more specific with experience. Or to counter that, I may be able to detect a sudden shift, a sudden change between this. And I might be able to infer from that in fact, this is a new neuron that suddenly has appeared that has coding properties maybe in a similar area to this previous neuron. So I'm not sure that I have an answer that you can always tell whether or not you're doing this, but I think these approaches give us new ways to potentially assess whether or not you're recording the same neuron and whether its receptive field properties are changing through time.
Cool. Thank you very much. I think you solve a huge problem in biology because, so I would like to ask you the question. All the time the referee tell us, okay, because we have to move the electrode, because that we didn't mention to the public here is we can't leave the electrode on the same place because we have a glial cell around. So that means we have to move the electrode. And sometime when I'm moving the electrode, I still have the same signature of the neuron, but I can't assume that is the same. And so the referee all the time told me, you can't say that you follow the same neuron.
Yeah.
I think it's possible with your tools.
Yeah, so it might be possible to track it. At the very least, what you could say is you could say this neural population is coding with the same properties. And so, even if I don't know that, say, this neuron here and this neuron here are the same, I can say that the population as a whole still seems to have a neuron with, similar waveform shapes that's coding in the same way as it was before. And so if I'm discussing it at the population level, I can talk about kind of maintenance of information at the population level. There's maybe potentially another step, but there's potential here in also saying, well, in fact, I'm confident that this was a neuron and that neuron has just shifted through time.
Thank you a lot. Other questions or comments? I have a few because it was really, really interesting. First of all, so I understand that in the end you want this closed loop detection, which means that you already know the kind of path the rat will be taking in the maze. So you know already the succession that you want to see, and this helps you to predict who is going to code for what. But if you say you don't know the maze, or you kind of try to guess what is the succession that the rat is making, could you kind of infer also the succession of the place on top of everything else? Or you really need to know in which sequence they are going to be?
Yeah, so in general, we've applied these types of methods in less structured environments. So in open field environments, or in each of the experiments I'm showing here, these rats are very well trained. And so the receptive field structure isn't changing very much. Or the hope was it wouldn't change very much, even though we found these changes. We've also applied these in kind of open field, less structured tasks, and when the animal is first introduced to these types of environments. And in those cases, we always need the ability to build a receptive field model, in this case, a population receptive field model that relates things like the animal's position and the spike waveforms to the timing of events. But in those cases, we have been able to use these methods to, say, decode with high accuracy the animal's movement through an environment. It's really this structured of alternating left, right. What that really helps us with is in these replay events to be able to disentangle these kind of leftward versus rightward trajectories. And in the cases where we're trying to predict actual movements, we're able to use them to predict right and left turns before the animal actually makes a turn. And so I think these methods as a whole are largely applicable even in less structured environments and less structured tasks, though in particular, this selection between outbound and inbound really relied on the structure of the task. Another thing we've done in the less structured task is try to determine whether or not these replay trajectories are coherent, whether they represent continuous mental exploration, or whether they're more fractured, whether they jump between environments. And that, again, doesn't require as much structure, this type of left-right alternation in this task.
Okay, thank you so much. And another question for maybe something which would be much more general than hippocampus and position coding. Would you be able to incorporate to the spike sorting other relation, like, for instance, connectivity between the neurons that would help you to decode, well, to spike sort the whole thing by adding those correlations between the spike trains or stuff like that?
Yeah, that's a great question. So, over the past 10, 15 years or so, classic univariate or multivariate point process models looking at multiple single units that are spike sorted have done a lot of work trying to understand connectivity between neurons and how using point process models can help you understand, pairwise relationships between neurons and how those adapt through experiments. We believe the same is possible with these joint mark intensity models, but we haven't actually implemented them yet. There's an additional challenge in trying to figure out how to represent, if you're not separately spike sorting, how to represent correlation structures between these types of clusters. So we can imagine what it would look like. So you can, for example, look at one of these clusters here and imagine that each of these kind of blobs represents a neuron, and how a spike in one of these blobs would then decrease the intensity in its own blob temporarily because of the refractory period, and maybe with some time lag, increase or decrease the intensity of some of these other blobs. So it's certainly possible. It's actually an area of active exploration right now, but we haven't implemented those yet. But I think there's a lot of potential in adapting these joint mark intensity models to have that type of correlation structure.
Okay, thank you so much. Other questions or comments in the room? On internet, I don't think so. So thank you so much, Uri, for this beautiful talk. And
thank you very much.
We have a half-hour break right now, and then we will come back for Thibaut Taillefumier and Robert Casse. Thank you.